All right. Thank you all for coming in person here at Hatfield for Angelina's PhD defense. And thank you to all of those attending online. It looks like we have all committee members present, either in the room or on Zoom. So I just wanted to take not too much time because Angelina has a really full presentation. But many of you may not know that Angelina first came to OSU as an REU in 2017. She was an undergrad at Humboldt State, working with Dr. Paul Bordeaux on PyZaster, a project in which ultimately she was considered enough of a collaborator that she's a co-author on the publication from that work. So co-author as an undergrad. As I was preparing these statements, I looked back at Paul's letter recommending her for the REU, and he wrote, "Even as a freshman, Angelina stood out as being organized, motivated, and equipped with a plan for what she wanted to do next, parentheses, attend graduate school and eventually teach." And so I think absolutely organized, motivated, and equipped with a plan. All of you who know Angelina know that that like describes her to a T. For her REU here at Hatfield, she investigated the effects of the 2014 to 15 marine heat wave, known as the blob, on the caloric density of Kringon shrimp. So that became her introduction to considering about temperature effects and marine heat waves, which you'll hear a lot more about for the next hour. That project continued on with a second REU the following summer, and then ultimately, when Angelina did come back here, we put that together into a publication. So written as a PhD student, but from work, again, she did as an undergrad. So while Angelina's undergraduate experiences were focused on invertebrates, she fell in love with seaweeds, as one does as soon as you learn about them. And she went to Cal State Fullerton in 2018 to pursue a master's degree with Dr. Jen Burniford, studying the impacts of Egregia menzii on intertidal communities in Southern California and Northern Washington. And you may note that there's a state in between there that perhaps should receive some attention. After finishing her master's, or towards the end, she contacted me about returning to OSU as a PhD student and continuing her work on kelps. And as my first love is seaweeds, I was thrilled at the idea, but I warned her, I don't have funding to work on seaweeds, so if you come, this is going to be a really teaching intensive PhD. And she was like, no problem, that's what I want to do. I'm going to get the G-cut, which she did. And throughout her time as both a master's and a PhD student, Angelina has worked via her teaching, her committee service, and as a research mentor to increase access and inclusivity, particularly for underrepresented students. And so I feel like that's a major contribution. So she's here today to present the culmination of her research efforts here at OSU, which is just a portion of the contributions she's made to the department, the university, and the field of marine science. And Paul, I think you're online. I'd like to let you know that Angelina has achieved her goals of attending graduate school and eventually teaching, as last week she was hired as a full-time biology faculty at Jefferson Community College, one of the SUNY community colleges near Lake Ontario in New York. And I measured it this morning on Google Maps. It's 21 miles from the border. Staying at those northern latitudes, but now she's got a giant freshwater ocean near her. So we'll see what comes of that. So thank you, Angelina, for your commitment to this project, to the lab, to OSU, and congratulations on this milestone.
Thank you. Okay, let's see. Here and then here. Okay, perfect. Thank you. Okay, we're just going to jump straight into it because I have a lot to say today. So for the talk breakdown, I'll be going over the 5 chapters of my dissertation. So chapter 1, we'll just go into introduction. Chapter 2, I'll talk about kelp thermal tolerance. Chapter 3, I'll talk about surf grasses and kelps, mostly related to their thermal tolerance, their canopy cover changes, and their community effects. Chapter 4, we'll circle back to kelps, and we'll look at their canopy fluctuations and the community effects associated with that. And then I'll wrap up with some final conclusions. So I want to introduce a term, foundation species, which was originally coined by Paul Dayton. It broadly includes organisms that provide habitat, resources, and support diversity. A good example on land would be redwoods. They provide the majority of the canopy in their habitats or their land forests. You might be less familiar with our underwater forests, but we have many foundation species that create nice forests underwater, including our grasses, which are our true plants, and then also our kelps, which are brown algae. And I just want to introduce some headlines if you haven't been watching the news too closely because it's scary on there. Here's just some recent things that we've been seeing here in the Northeast Pacific. For instance, kelp forests are vanishing along California's coast. We're losing bull kelp in the Puget Sound. And then the Nature Conservancy really highlights that we are losing a forest, our vanishing forest. And some recent literature is also showing, of course, that we are losing our iconic kelps, especially around the Monterey Peninsula, but there is some hope in other places like Oregon and Mexico. So we're going to focus in on Oregon today, and this is a report from the Oregon Kelp Alliance. This is their 2024 report, focused around Nereocystis, the bull kelp. And this is showing the whole coastline for Oregon, and I want you to highlight the orange labels are the ones that are showing loss in the last decade. So between 2010 and 2022, we're seeing pretty substantial loss north of here and also south at some of the southern sites. So we have seen substantial loss of some of our subtidal kelps. And there's many factors that are driving these losses, but one of the most focused one is temperature. And so if you look at this, you can see that every year since 2014, we are experiencing warmer temperatures compared to the mean. And on top of just those changes, this ocean warming up, we are also having events called marine heat waves. These are short periods of time, days to months, where we have unusually warm conditions in the ocean. And there are many reported marine heat waves globally, but the most notable one, or the one that was the longest in most recent years, was given its own name. We call it the blob. And this is just showing you kind of the temporal change through time of that from the NOAA fisheries. And you can see that if you look at these top figures, how the temperatures have shifted and how they moved around the Northeast Pacific through those years. But essentially 2015 and 2016, we sustained pretty warm waters. And there's been a lot of research around the blob, and these are just some of the negative effects we've seen. We've seen shifts in copepod assemblages. In other parts of the world, we've seen cellular damage in our coral reefs when marine heat waves are happening. And here in Oregon, in the Henkel Lab, we've seen reduced caloric content of shrimps related to that marine heat wave. And on top of all of this, in a recent analysis that was presented at the International Seaweed Symposium, it was highlighted that the majority of our physiological kelp data is related to the two main subtidal kelps, which is Nereocystis, the bull kelp, and Macrocystis, the giant kelp. So the majority of our physiological data is related to these two subtidal kelps. And again, highlighting that we have seen quite a bit of reports of their loss. Here's just an example from another study showing blade elongation or growth across different temperatures. And you can see that Nereocystis does pretty well between 10 and 15 degrees Celsius. But if you go above or below that, you do see a reduction in its ability to grow. And so they are threatened by temperatures. But today, I'm not going to talk to you about the subtidal. I'm going to talk about the intertidal. The intertidal is the place on the coast that becomes exposed as the tides go out. It is considered to be an extreme habitat because of the conditions that are there, including daily tidal shifts, extreme temperature fluctuations, including both water and air, and all of these other conditions too. But this is also a really diverse habitat, so we know that organisms living here are well adapted to these conditions. But what we don't really know, and where a lot of focus of research is now, is about the increasing pace of environmental change that we're seeing. How is that going to influence our intertidal communities and the interactions that we see there? So I'm going to be talking to you about 3 macrophytes today. The first one is Egregia menziesii, commonly referred to as the feather boa kelp. It is a foundational, and it is a perennial, so it can live for many years. And it does have a pretty large geographic range for our intertidal kelps. It's found all the way from Alaska down to Baja California. I'll also be talking to you about Hedophyllum sessile, which is our sea cabbage kelp. Also foundational, also perennial, does have a slightly shorter geographic range. It's found from Alaska down to about Monterey County in California. And then the last macrophyte I'll be talking to you about is Phylospadix, or our surf grass, also considered a foundational. The main difference here is that the first two are kelps, those are brown algae. That last one is a true flowering plant, and we have 3 species here in Oregon. And then it has a very similar range to Egregia, Alaska down to Baja. So what do we know about thermal tolerance? This first figure on the left is just from the Southern Hemisphere from the Wernberg lab. The main takeaway I want you to see is that they looked at 3 different macrophytes, and you can see that each one had a slightly different thermal optimum, and then on each side, how their photosynthesis decreases kind of varied within each species. So we know that thermal tolerance is really species specific. We also know that thermal tolerance limits growth and survival. And then thermal limits can lead to range contractions, and they've been seeing that in the Southern Hemisphere. So here in Oregon, we know average Oregon intertidal water temperatures are about 10.5 degrees Celsius. And from some other research coming out of integrative biology, we have seen that marine heat waves like the blob have elevated our intertidal temps up to 16 degrees Celsius in recent years. And you can see that by these boxes highlighted here. So we are warming up in Oregon. And there has been some other great work done, especially in the Sailor Sea at Friday Harbor Labs, where they looked at 51 macrophytes, which is a huge lift. And they reported the kind of thermal limits for quite a bit of species. And that included Phallospadics, which they said the upper limit was 25 degrees Celsius. Egregia had an upper limit of 18 degrees Celsius. And then Hedophyllum was in the lowest group, and it had an upper limit of 15 degrees Celsius. And so some things we don't know are the thermal limits the same outside of the Sailor Sea? If you're unfamiliar with the Sailor Sea, it's kind of just left of the Puget Sound, in between Vancouver Island and the upper northern Washington Islands. And so that's quote unquote protected compared to the open coast of Oregon, right? So do these thermal limits vary if you're out here? And then how does the temperature maximums affect the other things outside of net photosynthesis? So such as respiration, caloric density, growth, and can they survive longer than a week at their upper thermal limits? And then finally, I've told you that all of these 3 species are considered foundational, which means they should be very important to their habitats and the communities around them. So I will also be talking about and trying to answer how are the understory communities affected when the canopies are lost or reduced. Before I get too much further, I do want to do quick acknowledgments for everyone that helped me do all this work. You're going to hear me say we quite a bit instead of I, because I did not do the majority of this work alone. So I want to highlight all the undergrads and the grad students that help make all of this work possible, including my interns, Justin, Riley, Martin, and Kalia. All right, let's get into the data. So chapter 2, I'm going to be focusing on Egregia only, and I'm going to be talking about the impacts of manipulated light, temperature, and nutrients on its performance. And this paper is currently in review with Botanica Marina. So the objectives, there were 2 main objectives for this work. The first one was to examine the interactive effects of light, temperature, and nitrate availability on the performance of Egregia. And performance is kind of an umbrella term for their net photosynthesis, respiration, growth, and caloric density. And then I also wanted to look at and quantify the caloric density year-round for unmanipulated Egregia, but I will only be talking about this first objective today. So what did I do in the lab? We did 4 lab experiments from 2023 to 2024. All of our individuals came from Boiler Bay, Oregon. We drove them quickly back to the lab here, and they were immediately moved into tanks. They were cleaned, and then they were moved into their experimental tanks in the experimental seawater facility right across the street here within 24 to 48 hours of collection. The temperatures and the variables we were examining for each experiment did vary, so I will go over that on the next slide. But I first want to go over what the setup was for the experiments, because they were pretty similar. So each individual Egregia had its own tank, and then the water is flowing in here, and they had bubblers underneath. So they're kept in their own tanks. They're spinning on all the days that we weren't taking measurements. And then the system is recirculating, so the water flows out. There's a sump right here, and then it flows back into it. And then the lights are on timer systems up here, and then each setup had a tarp to kind of limit the additional light from the building and outside. And then basically what we did was we tracked their physiology, and so we looked at their net photosynthesis, their dark respiration, their growth, and their caloric density. And we looked at the net photosynthesis and dark respiration specifically with a Hanna dissolved oxygen probe. And when we were taking measurements, we would trap the individuals in these bottles, and they would stay in those bottles for the day that we were measuring them. But the rest of the time, they were free spinning in their tanks. And today, I'm just going to be talking to you about net photosynthesis and dark respiration. So here are the 4 experiments. I know it's a lot, but I want to focus on when we did these. So we had a May 2023, July 2023, April 2024, and July 2024. So essentially 2 springs and 2 summers, 1 year apart. And they were all new sets of individuals every experiment. They ran for slightly different days, depending on the experimental conditions. And then here are the factors that we were investigating. So for the first spring, we looked at temperature and light. So we exposed them to 14 and 16 degrees Celsius, and then shaded half of them. For the first summer, we just looked at temperature, but we brought the temperature up a little bit. So we did 14, 16, and 18 degrees Celsius. The second spring, we just looked at temperature. We did 13, 14, and 15. And then that final summer, we dropped the temperature quite low. We just did 11 degrees and 13 degrees Celsius. And then we added nutrients. And so basically, temperature, we were always investigating. And then we added some additional factors depending on the experiment we were in. And for light, light levels are important for autotrophs, so reducing it here should negatively impact them. That's what we were expecting. And then for that last experiment with nutrients, that should improve their performance if it's a limiting factor. And we do have periods of low nitrates here in Yaquina Bay, which is where our water for Hatfield is coming from. Okay, let's get into the data. So I'm going to orient you to these figures, and then all the figures will be set up the same. So we're going to have net photosynthesis always at the top, respiration always at the bottom, and then it will tell you the date or month and year of this experiment, and then the factors that we were looking at. So this first one we were looking at temp and light, and then the symbols indicate the mean for that treatment group, and then the N will be down here at the bottom. So for this first May 2023 experiment, main takeaways are that there was no effect of reduced light. We reduced the light by 50% here, but that we didn't really see any negative effects of that. And then higher temperatures did increase respiration. And if you look here on day 0, 7, and 14, you don't see big differences in respiration. But by day 21, you do see more negative results for the group that was at 16 degrees Celsius. But to highlight, we saw increases in respiration, but no change in net photosynthesis, which means net photosynthesis is compensating for the increase in respiration, or there's more photosynthetic capacity there. So thus, gross photosynthesis must have increased for this experiment. I'm going to show you the second spring. So we'll go by spring and then summer. So as a reminder for this experiment, we looked at 13, 14, and 15 degrees Celsius. And I want you to highlight, I'm going to highlight here that on day 0, 6, and 12, you don't see big differences in net photosynthesis between these treatments. But by day 18 and day 24, you do see separation with the hottest treatment, or 15 degrees Celsius, maintaining the highest net photosynthetic rates. And then in terms of respiration, we didn't see too much effect of these temperatures on respiration. So main takeaways for the spring experiments, we had no mortality. So everybody survived between 13 and 16 degrees Celsius for up to 21 or 24 days, depending on the year we did this in. We also saw consistent performance at 14 and 15 degrees Celsius, and we saw the lowest performance at 13 degrees Celsius, or the coldest temperature. And a thing to highlight is that the differences took weeks to see. So if we had stopped this at day 7 or day 14, we would not have been able to pull out these results. So now we'll switch to the summer experiments. And I just want to highlight as we transition from spring to summer, based on the spring 2023, 14 and 16 did not seem to be too stressful for agresia. So for the next summer, we added 18 degrees Celsius. We just assumed that, adding 2 degrees, we'll see what happens. And you can see by the lines, so each individual is its own line here. You can see that the lines come to a halt before the end of the experiment, and that tells you that we had mortality in our summer experiment. And so you can see that quite a bit of individuals did not make it to the end of the experiment. And so here we had mortality at all temperatures. We even had mortality at 14 degrees Celsius, which is much lower than their thermal limit has been proposed to be. And if you focus on the very last day here, day 28, for both photosynthesis and respiration, we had 3 individuals at 14 degrees Celsius that survived, and then we had 2 at 16 and 18 that also survived the full 28 days. And if you look at their performance on that day for those surviving individuals, they are performing if the same, if not a little better, than their initial measurements before they entered these treatments. So good survival or good performance for the survivors. And so this is just a reminder, this is a nice healthy egregia in the field. And this is what it looks like in the tanks when they're very unhappy. They disintegrate or they start basically deteriorating in their tissues. So they're quite unhappy at certain temperatures. And so for July 2023, this is why we did not push them any higher. We chose to use 11 and 13 degrees Celsius here because we do want them to survive so that we can actually take measurements of the tissue at the end. We couldn't do that with our summer individuals because we lost the majority of them for July 2023. So for the second experiment, we did 11 and 13 degrees, and then we added nutrients. And we hypothesized that adding nutrients would improve performance at both temperatures. But you can see that lower temperatures in general did reduce their performance compared to the higher temperatures of the previous summer. And nutrients as a whole did not increase photosynthetic performance substantially. So I want to highlight what the conditions were like when we took these individuals out of the field. So here I'm showing you buoy water temperature off of Newport. Here's intertidal hobo water data, and then here's the temperature of the Yaquina Bay water as it gets pulled into Hatfield here. And I've highlighted the 2 springs. So what I want you to see is that for the offshore, we're about 9 degrees Celsius on average. For the intertidal, we're about 11. And then Yaquina Bay is between 11 and 14, depending on the day. And then for summer, I want you to notice how much colder it is. So in the buoy water temperatures were about 7 degrees, so almost 1.5 degrees colder. And the intertidal is almost 2 degrees colder on average in the summer. And then Yaquina Bay can fluctuate quite a bit. This second summer was pretty hot. That was also one of the hottest days that we had in Newport that summer. So it's very warm sometimes in Newport. And so what I want to highlight here is that for the chapter 2 conclusions, the timing of our El Niños and our heat waves really matter. Our summer individuals are more susceptible to warmer water temperatures than the spring individuals. And also what's really important is the duration of our warming events. It's been proposed that the upper limit of egregia is 18 degrees Celsius, but I also saw mortality as low as 14 degrees Celsius. But we also saw individuals at all of these temperatures persist all the way through and perform well. So the duration of these warming events can really impact population survival. Okay, chapter 3, we're going to switch now and talk about higher thermal tolerance and increased canopy cover after disturbance in surf grasses and kelps. And this paper is currently in review with aquatic botany. Okay, so we're going to focus on egregia again. As a reminder, it's a foundational and it's a perennial. And we're also going to talk about Phyllosphadics, also foundational. Main difference here is that egregia is a kelp, Phyllosphadics is a grass. And they have very similar geographic ranges. Both genera are found all the way from Alaska down to Baja California. And the objectives of this chapter were to first quantify thermal tolerance of both species. Second, quantify understory differences. How does the understory look different between these 2 dominant canopies? And then third, quantify canopy recovery after canopy removal and understory consequences. So again, we did 2 lab experiments for these species together, and we did these in 2025. For lab 1, this was done in April 2025, and we exposed them to either 13 degrees Celsius or 15 degrees Celsius, and this one ran for 28 days. The setup was very similar, but instead of having only egregia, now it's egregia in one tank, next one surf grass, next one egregia, and they just bounce back and forth. And then for the second experiment, we did that one in June 2025. The control or the starting baseline was pretty hot. We did 15, average between 15 and 16 degrees Celsius. And then we had a heated treatment where we heated 1 degree Celsius additional every single day. So we were heating it basically constantly. And we did a bunch of physiological monitoring. Today, I'm just going to be focusing on these three: gross photosynthesis, net photosynthesis, and dark respiration. And here's what they look like trapped in their bottles. Okay, so I'm going to talk to you about the first lab experiment, 13 and 15 degrees Celsius. To orient you to these graphs, we're looking at metabolic rate here on the Y, and then the day on the x-axis. And then these left panels are gross photosynthesis, middle panels are net PSN, and then right panels are respiration. And then I've split the species up, so Egregious is on the top, Phallospadex is on the bottom. And then in terms of treatment color, if they're red box plots, those are the 15 degrees Celsius compared to the 13 degrees Celsius. What I really want you to take away from this graph is that the metabolic rates did not vary by temperature for either species. And if you look at the gross PSN here, you can see that the gross photosynthetic rate was elevated for Egregia for days 7, 14, and 21. As we saw in the chapter right before, it does perform a little better at the higher temperatures like 14 and 15. So we were expecting that, but by day 28, it started to match 13 again in terms of its performance. And then we know that Phallospadex is robust and can withstand up to 25 degrees Celsius. So both species are capable of withstanding kind of the blob temperatures that we were experiencing or that we've already previously experienced. So for the next experiment, we looked at how much higher can we heat them up. So as I said, the control here, or the like bottom temperatures, were the 15 and 16 degrees Celsius treatment. And then the heated treatment got an additional 1 degree every single day. So by day 7, we were at 23 degrees Celsius, which is quite elevated for Oregon. And I want to highlight that everything is the same in terms of metabolic rates and the x-axis here. But I really want you to focus in on respiration for Egregia here. You can see that by day 2, which is when the heated is reaching 18 degrees Celsius, which is considered its upper thermal limit, you can see there's a significant difference in respiration. The respiration is very high, and as it's heated, the separation between the treatments starts to grow. And then you can see by day 7, we had one individual that's respiration was through the roof. And you can imagine the next day I came into the lab, it was fully disintegrated. So we stopped the experiment because we had reached a mortality event. If you look at Phallospadex, not a lot of differences happening, even if we're heating them all the way up to 23 degrees Celsius. So metabolic rates did not really vary for Phallospadex between these treatments. Okay, so now let's talk about what we did in the field. So we did a big field study here in Oregon. We had 4 sites. So we're right here at Hatfield. We had Fogarty Creek and Buller Bay, which are north of us. And then we had Bob Creek and Cape Blanco, which are south of us. And we used a block design. And so what that looks like is right here. So essentially every block had 9 plots. And so the first 3 plots here had Egregia only. The second set of plots had Phallospadex only. And then this bottom set had mixed. So it had both Egregia and Phallospadex present in the plot. And then we did full, where we had multiple blocks at each site. So Fogarty Creek had 4 blocks, Boiler and Cape Blanco also had 4 blocks. And then Bob Creek was our only site that had less blocks, and it also didn't have areas where the 2 species mixed. So we're missing this bottom category right here. And then we did community surveys. So we looked at algae percent cover and sessile and sedentary inverts and percent cover. And then we also looked at mobile inverts, but I'm only going to be talking to you about the things that were attached today. So in terms of timeline, this started in June 2023. We did full community surveys, and then we did our destructive event. And what that looks like is after we did our full surveys, a third of them, or 33%, were kept as a control. 33% were sheared. And by sheared, I mean we just completely sheared off all the canopy, but we left the holdfast and the rhizomes in the plots. And then we had a full removal category where we removed everything out in terms of the canopy. And so that also happened in June 2023. We only had a single disturbance event. And then we surveyed every 6 months. So we always surveyed in the summer and the winter. And then we completed this in June of 2023, so basically 24 months after initial. Today, I'll just be talking to you about June 2023 and June 2025, so kind of that 0 and then 24 months. For statistical analysis, I just want to highlight that for the diversity metrics, we looked at species richness, evenness, and Shannon diversity indices, and those were all calculated using the algae and the sessile invert data. And then we did ANOVAs and Tukey's to analyze the canopy changes and the diversity metric differences. We also looked at the community in terms of multidimensional scaling and Permanova, but I'll just be talking about these top two today. So I first want to orient you to what the canopy looked like before we did any type of manipulation. So here is Egregia percent cover, and then here is Phallospadix percent cover. And then I've separated the 4 plots. The northern plots are the top 2, and the southern plots are the bottom 2. And the colors indicate the canopy, like the main canopy on top. So Egregia is in red, Phallospadix is green, and then we have these gray ones that are that have both, essentially, so mixed plots. And then the symbols indicate what treatment they got after we took this data. And so you can see that we have our plots that just have Egregia, our plots that just have Phallospadix, and our plots that have in between. Now, what does it look like 24 months after manipulation? So all the axes are set up the same, and I've kept them the color that they originally were, so you can track some of them. But I'm going to walk you through what I want you to take away from this. The first thing I want you to take away is that 86% of our Phallospadix dominated plots were still Phallospadix dominated after 24 months. And as a reminder, 33% were kept as a control. So a third of them we sheared, a third of them we removed, but after 24 months, 86% are still Phallospadix dominated. For Egregia, 36% of the original Egregia dominated plots were still Egregia dominated 24 months later. And this is regardless of the treatment that they got. Specifically, if you really look at Boiler Bay, we had a nice amount of Egregia in the beginning, and we lost all of our Egregia on the bench where these transects were running. And that's regardless of being a control, a shear, or a removal. Egregia just went away at that one, the right side of Boiler Bay. And then you can see, if you look at some other ones, we had like some the triangles indicate our removals. And you can see that some of the triangles, they were fully removed 2 years before, and now they're sitting at 100% or 75%, things like that. So this dominance, that's 36%, is regardless of the treatment that they got. And then finally, with the mixed plots, I've highlighted what the mixed plots look like in the beginning. But by June 2025, only 24% of mixed plots were still mixed. And if you look at where the gray ones are in this second figure, you can see some of them have moved to be just Egregia dominated, and some of them have moved to be just Phallospadix dominated. So there was substantial shifts there. Now I will tell you about diversity. So I'm going to show you Shannon diversity here on the y-axis. X-axis are the 3 different canopy types we started with. So we have Egregia, mixed, and Phallospadix, and then we have the 4 sites. And then the numbers above indicate how many plots we had within each canopy type. What I want you to take away from this is that at Cape Blanco and at Boiler Bay, we saw no differences between Shannon diversity across any of the canopy types. So basically this similar diversity, regardless of if you're under Egregia only, Phallospadix only, or mixed. At Bob Creek, Phallospadix was elevated in its Shannon diversity, so the understory had a higher Shannon diversity in the Phallospadix plots compared to the Egregia plots. And then at Fogarty Creek, it has an inverse relationship. So the Phallospadix plots had the lowest Shannon diversity compared to the Egregia or the mixed plots. Now I'm going to show you 24 months later, but first I have to introduce a new category. Our none category is now in black here. And these are our plots that have neither Egregia or Phallospadix now 24 months later. And just as a reminder, all the numbers here equal the same number of plots that we had here. So we're still monitoring the same plots. They have just changed with their dominant canopy. And so what I want you to take away from here is that at Cape Blanco, Bob Creek, and Boiler Bay, there were no differences between the Shannon diversity for any of the canopy types, including that none category. Fogarty Creek did maintain its inverse relationship, or the negative relationship with the Phallospadix plots for its Shannon diversity compared to the other 3, even when you introduce that neither category now. So I've told you that diversity didn't change or wasn't super different between at least 3 of the sites between these canopy types. But it is important to note that the understory identities do vary by the dominant canopy, and this varied by site. So the pattern wasn't the same for every single site, but the main general take-homes are that if you pull away a bunch of egregia, if you lift up all of egregia's fronds and put it to the side, this is the majority of what the plots look like, especially especially in our macrophyte-dominated capes. For instance, you see quite a bit of red algae, quite a bit of fleshy reds. Whereas if you move away all the phyllospadix, or if you can dig through to the bottom, you get more inverts, and you also see an abundance of coralline crusts underneath there. So the identities are different, but diversities were the same. So the chapter 3 conclusions are with some warming, both egregia and phyllospadix will be okay, but if we have rapid warming, that will threaten egregia populations more than phyllospadix populations. In terms of disturbance, after we did our big disturbance event, almost all phyllospadix plots were able to recover after 24 months, regardless of the canopy manipulation type. And then in terms of diversity, at 3/4 of the sites, diversity was not different between phyllospadix and egregia-dominated plots. But the identities were, so the present species do vary by the dominant canopy. Okay, chapter 4, last data chapter. We're going to talk about the sea cabbage, or Hedophyllum sessile, and we're going to talk about fluctuating canopy cover and the impact on community assemblages and productivity. So I want to highlight a classic manipulation study, Dayton's 1975 paper. This was done in Washington. It included a few species, but there was also a focus on Hedophyllum sessile. It's been cited over 900 times. And the main highlights from this are that the relationship between Hedophyllum and understory species can vary by environmental conditions at the different sites. And he did a classic removal, so you see quite a bit of, this is just a subset of his plots, but you can see that he did removal and then basically tracked the understory species through time. But I highlighted here his controls to show and emphasize that Hedophyllum canopy changes with time without being removed. And it seems like there's a pretty strong seasonal fluctuation here. And so the objectives today for this chapter were to revisit Dayton's 1975 and really focus on quantifying Hedophyllum's canopy cover and understory communities for 8 seasons. So we did this in 2023 to 2023, or 2022 to 2023. And we had no manipulation. So this is just pure 2 years of observation tracking the canopy and its understory communities. And then the second objective was to assess recovery of Hedophyllum canopy cover and understory consequences after canopy manipulation and loss. And the third objective, similar to the chapter 2 egregia study, was to quantify the caloric density through time. I'll only be talking about the first 2 today. Okay, so we did another Oregon field study, and we did this one at Boiler Bay, so just north of us, and Seal Rock just south of us. And we also did a block design here, but it was different from the chapter I just talked about. For this block, we used canopy cover, and so we essentially had one plot, started with 0%, 1, 25, 150, 75, and 100%. So that would be one block, and then we had 5 replicates at each site. So for a total of 50 plots between the 2 sites. And we did, what that looks like here is that you can see this is one plot here. It has one individual Hedophyllum, very little canopy cover that it's providing. Less than 10% falls in that 0% bin. And then on the right here, you can see this is many individuals of Hedophyllum. They're taking up quite a bit of space and providing a lot of cover, falls into that 75% bin. And then we did community surveys. So we did algae percent cover, sessile and sedentary inverts, and we also looked at mobile inverts. I'll just be talking about the things that are attached today. And then in terms of observation, we started this project in February of 2022, and then we did community surveys every season for a total of 8 seasons. And then we did a destructive period where we manually removed canopy in February of 2024 to simulate kind of a similar removal. study, and our plan was to keep things at a certain level. So we anticipated going back and sharing it back again. But when we went back a month and a half later, we actually had even more loss than we were anticipating. So we had quite a bit of distal bleaching, which leads to tissue loss. So we had lost even more canopy than we were anticipating. So we did not do another destructive event, but we count this as more canopy loss. And so we did our final community survey in August of 2024. And so I'm just going to show you the canopy cover data first, and I'll show you all the observational year stuff first before I show you the post manipulation. And so here's Heatophyllum canopy cover, and then here's the 8 seasons of data collection. Each one of these lines is one plot through time, and then the squares are the averages for that month and year. And I've separated the sites, and hopefully you can see that there is a seasonal trend with an increase in our canopy cover into the summer, decrease fall into the winter, and then increase again in the summer. And we saw that at both sites. So overall, we have higher canopy cover in the summer compared to the winter. I'll show you our diversity data. So we looked at richness, evenness, and Shannon diversity. And I'm just going to be showing you evenness today because it had the strongest response to canopy cover. So here on the y-axis is evenness, x-axis is canopy cover. I've separated the sites, so boilers on the left, Seal Rock on the right. And then I've separated the seasons out here. As a reminder, we did 2 years of observation, but I don't separate the years here because we did not see differences between the 2 observational years. And main take-homes are that if you look at Boiler Bay, you can see a positive relationship between evenness and canopy in the spring and the summer, so these 2 middle seasons. Whereas at Seal Rock, you see that strong-ish, R-squared, positive relationships at the winter, the spring, the summer, and the fall. So all 4 seasons, you see a positive relationship between these 2 variables. But for Boiler Bay, in the winter and the fall, it's mostly flat, if not a little negative. And then I'll show you algae. So these are just the algae assemblages. This is all the algae that's attached within our plots. And I'm going to show you Boiler Bay and Seal Rock. And what I'm showing you here are the bootstrapped Bray-Curtis similarity matrices. And now I'm showing you the bins. So what I want you to take away is that in that block design, we had 0% bin, which is shown by our dark blue color. And then we had 100% bin, so that's our teal color here. And then red is 25%, green is 50%, and then pink is 75%. So now the canopy cover is pushed into bins. And what I want you to see is that the 0% separates out, 100% separates out, and then we have a clustering of the 25, 50, and 75. And we did see this at both sites. And when you look at this, if you look at canopy bin as the explanatory variable with the permanova, there are significant differences between these groups in terms of their algae assemblages. So the algae with 0% is different from the groups that had 100%, or different from all the other groups, and then 100% is different from all the other groups, and then these middle groups were not different from each other in terms of their algae assemblages. So 25, 50, and 75%, very similar in terms of their algae communities. And then very similar results at Seal Rock. The main difference is that the 100 bin and the 75 bin were not different from each other. So a little bit more overlap in their communities there. Now I will show you the post manipulation and post loss. So I've highlighted here where we did our first manipulation, and then you can see at both sites that the canopy is pretty reduced in our plots in February, and then it gets reduced even more in May, and that's caused by the distal bleaching and tissue loss that I showed. But then by August 2024, you can see pretty substantial rebound in almost all of the plots, and the average for that for both sites basically rebounded back into that mid-range, or that mid-25 to 75%. And so I'm just going to briefly show you this, not go too heavy into it, but this is Seal Rock, and this is showing the 3 summers. So we have 2022, 2023, and 2024. And now we've pushed them into bins. So we have low, mid, and high. So we've basically grouped 25 and 75 together. And you can see that here is 2024 directly on top of 2023 or 2022. And this is for the 0% and the mid tier. So the assemblages even though its post-manipulation matched 2022. So we didn't see big differences in the summer following this because the canopy rebounded to that kind of mid-level. And so the chapter 4 conclusions are that Hedophyllum has predictable seasonal canopy cover fluctuations here in Central Oregon. So what Dayton saw almost 50 years ago, we still see today in his controls. And then the positive effect of foundational canopy is site and seasonal dependent, right? So the positive effect of having more canopy isn't always true, especially at Boiler Bay when you compare it to Seal Rock. And then in terms of presence, we saw substantial differences in the understory between the 0% and everybody else and the 100% and everybody else. And then when you looked at percent, if you were focusing on percent cover, that mid group, that 25 to 75%, those understory communities were pretty similar. So that means if you're switching or losing canopy, but you're maintaining that range, your understory communities are pretty similar. So you won't see big shifts. And then in terms of the post, we saw no understory differences post manipulation loss, essentially because canopy had returned to that acceptable range. So it maintained the presence that mattered. And so what does this all mean? In terms of, we know marine heat waves are expected to increase in frequency, intensity, and duration. Here's just a quick graphic to show you where we've had marine heat waves in the last 2 decades. We're having them globally, of course, and we're having some pretty extreme ones in many parts of the world. I'm just going to highlight one study from the Tasman Sea 2017 marine heat wave. They saw local extinction of their fucoid bull kelp, and what they saw was that the community slowly was replaced by alternative foundation species. And they monitored this post the marine heat wave for 6 years, and their fucoid did not return. And so they had shifted from this basically to being dominated fully by something else. And it kept that state after. And so it's important to highlight, to understand the post-marine heat wave effects, we must have prior knowledge of the community variability. And so if you've been watching the news, we have seen some preliminary data coming out of Scripps. This was just published April 21st, so just a few days ago. I'll highlight that this was 2025, here this darker line, and these are the 2026 days with the red indicating the day record high. And I'll highlight that in 2025, on that day in March, late March, we were at 15 degrees Celsius, and this year we're reported to be at 21 degrees Celsius. So we are seeing a marine heat wave develop off of the coast of Southern California. And so in terms of the field surveys that I did today, that I talked about today, I tracked all 3 macrophytes in the field to assess canopy effects on understory diversity and identities. And when we look at all 3, lower Hedophyllum canopy cover does equal less diversity, especially if you lose it entirely. Whereas for lower or completely absent Egregia, we did not see diversity change. Diversity stayed the same. And then if you see switch from Phyllospadix or to Phyllospadix from the Egregia or the mixed areas, we saw similar diversity, switching between those 2 dominant canopies, but we did see different identities. And then in terms of thermal tolerance, we already knew that Hedophyllum had the lowest reported tolerance of the 3, so it's definitely at risk as we anticipate more marine heat waves. For Egregia, rapid warming, like what we are just now seeing in Southern California, will definitely put the populations at risk for local extinction. And I'm going to highlight that the timing and duration of these events really matter. Summer populations are more at risk. We're at risk of the marine heat wave down south moving up, and the timing of that will really tell us if the Egregia populations are going to be suffering. Will it hit us now in the spring, or will it come later in the summer? And then for Phyllospadix, Phyllospadix does have superior thermal tolerance and canopy recovery, and thus it may become the competitive dominant. And when it becomes the competitive dominant, we might see what they're seeing in the southern hemisphere, where it will take over the space, and then it will basically not allow other things to move back in after the ocean cools back down. And I'll finish with my additional acknowledgements. I'd just like to acknowledge my marine botany inspirations. When I was an undergrad, I was able to take 2 amazing phycology classes back to back. I took my very first one with Frank Shaughnessy at Humboldt, and then I took marine botany at UW with Tom Mumford and Wilson Freshwater, and that was both of those just solidified that I was going to take this kelp path. So I thank them for inspiring that in me. I will also acknowledge all my funding sources that made all this research happen. Of course, I'll acknowledge Sarah. I had this whole thing about being an REU, but she already talked about it. So I'll just say thank you so much to Sarah for being my advisor and for taking me back into her lab. It's been almost 10 years of mentorship since I was an undergrad in 2017. So I thank you for all that time, and I know we're going to continue to collaborate. So thank you. And then I'll thank my committee members, of course, that have stuck with me through the years: Sally Hacker, Bruce Mengi, Alan Milligan. I'll thank all my GCRs: Suzanne Brander, Julia Jones, and now James Watson. I'll thank all of the members of the labs that we've collaborated with, like the Henkel Lab members, Mengi Lab, and the Hacker Lab members. Also, all my teaching teams. They made the experience here at OSU amazing: 22X, 20X, and 450. And then I'll thank all of the admin at Hatfield and Integrative Biology. It took a lot of manpower to get all these projects together, so I thank everyone, and I thank my family, and of course Kyle and our cat Aries. And I'll take questions.
Thank you, Angelina. We're going to use the mic even for questions in the room so people on Zoom can hear if we ask questions in the room. I will run the mic for the room, and Cinnamon will run the mic for online. So do we have any local questions? Matt.
Awesome job. What's the coolest kelp and why?
What's the coolest kelp? Definitely Egregia. Egregia is the coolest kelp because it has multiple morphologies and it has such a large geographic range, found all the way from Alaska to Baja. It's understudied, underappreciated.
And yet a fashion statement. Scott.
Great talk. I'm curious about the egregia loss at Boiler Bay in 2023. It coincided with the same time that there was this historic expansion of the bull kelp in that region, off fall weather, and that whole area was the most bull kelp we've seen in many years, which also could be thermally sensitive. But do you have any more insight into what was going on with that egregia loss?
Yeah, so Egregia did not disappear from Boiler Bay. It just disappeared from the right side where we did this project. So let's see if I have a good picture of what that looks like. So here, so this is, if you know Boiler Bay, this is the middle, right? The boiler's way over here. So this is the right side of Boiler Bay. So this is when we started the project, and basically all the brown is Egregia here. And yeah, 2 summers later, it was completely gone. But it was still fully abundant at the left side and like the more to the right. So I'm not sure if it just was not able to recruit because Phalosphaedix, just the primary canopy, grew so much. The next most common thing in our plots that had neither canopy was Mausiella and then Hedophyllum. So Hedophyllum did take up some space where it wasn't originally, which is interesting, but I did not record any bull kelp, even recruits. I did see them quite a bit in the bay during that summer, and right before too, but not in my plots. So I don't know if that would be impacting them. Yeah.
Tom.
Thanks, Angelina. Learning a lot about macrophytes today. So, a question about the diversity indices that you use, and I think you said you combine the invertebrates and the macrophytes together. And I'm just curious, where you didn't see changes in evenness or diversity, was one of those groups dominating the other one, or just something about like what the composition of those groups were compared to each other, and if you did delve in and maybe had an inclination that maybe one group was more variable than the other or more responsive to changes, your manipulated changes in the canopy.
Yeah, great question. Yes, we definitely looked at both separating algae and inverts for all the diversity metrics, and then we also combined them. In terms of where, like if you looked at my study sites, I'm really picking places that are macrophyte heavy. I didn't do any of the sites like around Cape Perpetua, like Strawberry Hill, because um, because I needed so much kelp to be able to get these plots done, right? So the algae, if you look at just the algae, it just dominates in terms of percent cover. And so when you separate out the inverts, there's really no strong relationships because there's very little in relation to like how much algae percent cover there is. So it was much harder to pull out the sessile inverts and try to find any patterns because there's so much macrophyte dominance. But where we did see it was definitely with the phallospadix plots. So we like separated out the invert and algae data there.
And just a little clarify, when you were doing those, were you including these 3 species in those diversities, or that was everything else except for those?
Yeah, these species are removed and they're holdfasts because their holdfasts also take up quite a bit of space. And if I'm targeting Hedophyllum, that would just skew the data, right? So yeah, we removed Hedophyllum, primary canopy, and holdfast from the analyses.
Cinnamon, anything on the chat?
Nope, lots and lots of congratulations, though.
All right.
Felicidades, doctora.
Hi.
So you're at the end point here now. If you could go back when you first started, is there anything that you would do different about your research? Or is there something new that you thought that you could have done differently?
Ooh. Well, I would apply for more money and try to get more money back in 2021. The biggest challenge of this whole thing was that I couldn't control the lower limits here at Hatfield. I needed way more money to have chillers. So I was kind of at the mercy of the building temperatures. So yeah, if I could go back, I would try very early on to get money to get chillers so that I could do like a bigger range of temperatures and include the lower temperatures as well and increase the temperatures. So, but would I have gotten the money? I don't know.
That was a great question. Thank you. Scott, you got more?
Maybe just to follow up on that, do you have thoughts on what you might expect about differences between sort of short-term physiology, respiration, photosynthesis based observations over the matter of weeks